gradient in the responding organs, leading to the differential growth. To elucidate a molecular mechanism of shoot gravitropism, Hidehiro Fukaki had isolated many shoot gravitropism mutants like this of Arabidopsis SGR mutant in Masao Tasaka's laboratory. This is one of the mutants. Oh, it's working? Oh, yeah. Yeah. This is one of the SGR mutants. So through the investigation of SGR1 and SGR7 mutants, Fukaki-san demonstrated that the endodermis is essential for shoot gravitropism. And in Arabidopsis, shoot endodermal cells and root columella cells contain amyloplast, which sediment in the direction of gravity. So there fits well, these are fits well with the starch-startless hypothesis, as you know well. And so we can conclude that these cells are gravity sensing cells called statocytes. And when I started my research on gravitropism in Masao's laboratory, a lot of genes involved in gravitropism had been already reported, and the molecular mechanisms of auxin-related process here had been extensively studied. And on the other hand, early process here, early process of gravity, early process of which occurs in the statocyte was largely unknown. So I focused on these processes. So I analyzed the rest of the SGR genes and found that normal function of the vacuoles and actin cytoskeleton are important for amyloplast sediment, normal amyloplast sedimentation in the shoot statocyte. However, the analysis of SGR gene did not reach on the signaling process. So we switched the research approach and we performed the transcriptome analysis focusing on the statocyte. And I compared gene expression profiles between the wild type and shoot gravitropism mutant defective in the statocyte formation. SGR1 scarecrow mutant cannot form the endodermis and show no gravitropism. And the EAL1 hypomorphic allele of SGR7 short root can form the endodermis-like cell layer, but it completely lacks the gravitropic ability in stem. So we expected that we can concentrate the genes involved in gravitropism by extracting the gene down-regulated commonly in both mutants. So as a result, we found LZY like family genes. But when I got this result in 2007, they are annotated as unknown proteins. So in the same year, 2007, the responsible gene for a well-known rice mutant, LZY, was isolated like this, and rice LZY exhibited reduced shoot gravitropism. And we found that weak sequence similarity between our unknown proteins and the rice LZY, and started functional analysis using the Arabidopsis. To date, mutants lacking the homologous genes were found in many species, and they showed gravitropic phenotype in both shoots and roots. In Arabidopsis genome, there are 7 LZY-like family genes, and sometimes you may be confused because each research group uses a different gene name. So I'm sorry for that, but here I would like to use our nomenclature. Okay, this is a summary of our study on LZY 1, 2, and 3. And LZY triple mutant exhibited normal amyloplast sedimentation, but failed to form the asymmetric auxin distribution in the responding organ. As this phenotype was similar to that of the rice LZY mutant. And what we had newly demonstrated using Arabidopsis is that LZY 1, 2, and 3 redundantly function in shoot and root gravitropism. And we also demonstrated that LZY gene mainly expressed in the start site, and also we had demonstrated that start site specific expression of the LZY gene was able to rescue these triple mutant phenotype. So we concluded that LZY genes are involved in the signaling process following amyloplast sedimentation and leading to the formation of the asymmetric auxin flow. And however, with regard to the molecular function of LZY, little information was obtained from their amino acid sequences. So we found that the LZY3 protein exhibits interesting intracellular localization in columella cells. As you can see, LZY3M cherry was polarly localized in the direction of gravity. And 30 minutes after gravity stimulation by upside down, LZY3M cherry was repolarized to the new bottom side of the cell. And at that time, amyloplast displacement was also observed, and indicating that a strong correlation between LZY3 repolarization and amyloplast sedimentation. Actually, these images were obtained using fixed and cleared samples because it was really difficult to perform live cell imaging of LZY3 in our transgenic lines, probably due to the low expression level. To observe the dynamics of LZY protein in living start site, we switched to the target protein. LZY4 shares redundant function with the LZY3 in root columella cells. With LZY4M scarlet, the ring-shaped signal and nearby linear signal were clearly observed in living columella cells. This ring-shaped signal was observed at the amyloplast position indicated by the plasmid marker, and the linear signal was merged with the plasma membrane marker like this. And the LZY family proteins have no detectable transmembrane domain. Instead, there are regions with a high basic hydrophobic score, a cluster score in all members, indicated by red lines. The regions with a high BH score may associate with the membrane lipids through the electrostatic interaction. So when positive charged amino acid in the region with a high BH score was substituted with glutamine, as shown here, only the plasma membrane localization was disappeared. And the mutated LZY4, this cannot rescue the triple mutant phenotype. This indicates that the LZY is associated with plasma membrane electrostatically, and membrane localization is required for gravitropism. So we investigated the relationship between ARG1 and ARG2 and LZY localization. Patrick Masson's group in Wisconsin-Madison demonstrated that the ARG1 and ARL2 encoding the J domain proteins are involved in the gravity signaling process in stat site, in root and hypocortex. In addition, genetic interactions between the ARG1, ARL2, and TOC complex components of translocon of plastid have been reported by their group. As you can see here, both amyloplast and plasma membrane localization of LZY4 were lost in ARG1 and ARL2 mutant background, suggesting that ARG1, ARL2 are required for intracellular localization of LZY, probably through their chaperone function. So next, to carry out the live cell imaging of LZY dynamics upon the gravity stimulation, we used a vertical stage confocal microscope. I had laid down this old microscope over 20 years ago, and confocal devices are added, gradually added, and it is not sophisticated no more, but it works. It still works. And these are time-lapse imaging of live cell imaging of LZY4M scarlet in root stat site. Before gravity stimulation, the pattern of the LZY4 signal didn't change during observation, although the amyloplast slightly agitated. And in contrast, after gravity stimulation, the amyloplast sediment to the new bottom side of the cell in about 5 minutes. And at similar timing, LZY4M scarlet signal began to appear on the plasma membrane of the new bottom side of the cell, and the signal gradually became stronger. We also found that the fluorescent signal on the plasma membrane, previous gravity side, quickly disappeared when the amyloplast left there. And similar results have been reported by Howden and the Hughes group. Next, we examined the relationship between the LZY4 on amyloplast and its polarization at the plasma membrane. And we used a photoconvertible fluorescent protein, MEOS2, and photoconversion of LZY4 on the amyloplast was carried out by irradiation with a 405 nanometer laser. Then the behavior of the photoconverted LZY4 was chased. Red signal was chased. And red fluorescent became visible after around the amyloplast just after irradiation. And the fluorescent intensity on the plasma membrane proximal to the amyloplast significantly increased 1 minute after irradiation. So I think this result suggests that the translocation of LZY4 from amyloplast to the proximal plasma membrane. Next, we'd like to know whether the position of amyloplast determines the LZY accumulation site on the plasma membrane. Whether the direction of whatever the direction of gravity is. And we performed two kinds of experiments. And first, lighten the amyloplast, let them float. And the second one is move some amyloplast by an optical tweezers. The first experiment was performed with PGM mutant. PGM is a starchless mutant with reduced gravitropism. In PGM mutant, as you can see here, the amyloplast without starch granules were floating and moving randomly, and LZY signal was observed at the amyloplast and the plasma membrane without polarity. So for the second experiment, we use a confocal microscope equipped with optical tweezers to trap and manipulate the amyloplast position. And in this case, the direction of gravity is toward the back of the screen. And after trapping amyloplast, the amyloplasts were put in close proximity to the plasma membrane. And then the signal intensity of LZY scarlet here significantly increased at the nearby plasma membrane, indicated as indicated in this graph. And these results indicate that the not gravitational force, but the amyloplast position determines LZY accumulation site on the plasma membrane. So based on these results, we've proposed a new model for gravity sensing. And the LZY protein translocates from the amyloplast to the nearby plasma membrane. And LZY proteins associate with the plasma membrane with the electrostatic interaction. In addition, the rapid disappearance of LZY from the plasma membrane distal to the amyloplast appears to allow gravity-responsive LZY polarity formation. And therefore, the LZY act as signal molecules, transmitting positional information of amyloplast, that is the direction of gravity. Then, what does LZY polarized at the plasma membrane cause next? So we isolated RLD family proteins as interactors for LZY proteins. The RLD family proteins are well conserved among the broad range of plant species, and RLDs interact with the LZY via the C-terminal BRX domain. And I don't show the data here, but we have demonstrated that RLDs are likely to regulate oxygen transport through the membrane trafficking, and also demonstrate that RLD 1 and 4 are involved in the gravity signaling process in start site. So this slide shows the intracellular localization of LZY3 and RLD1 in Arabidopsis protoplast. RLD1 GFP was in cytoplasm and punctate structure. However, when RLD1 is co-expressed with LZY3 in the same cell, most RLD1 GFP signal was co-localized with LZY3 on the plasma membrane, indicating that LZY3 recruits RLD from cytoplasm to the plasma membrane. So we also observed that RLD1 behavior with LZY4 upon gravity stimulation. We found that polar localization of RLD1 here and LZY4 at the plasma membrane was was observed at 15 minutes after gravity stimulation. So we examined each processes of gravitropism under the almost the same condition on the stage of the vertical microscope. And we confirmed that a repolarization of LZY occurred immediately after amyloplast displacement, followed by polarization of RLD1. And these events were observed prior to the formation of the asymmetric auxin signal, indicating that repolarization of LZY RLD is an important event in the gravity signaling process. So this is a part of a list of candidates for the lazy interactor resulting from IP mass spectrometry. And RLD1 you can find here, and GNOME is also interacting candidates. But we couldn't interaction between the lazy and GNOME in East hybrid system. So in root epidermis, most GNOME GFP signal was found in the Golgi and cytosol, but weak signal are also observed on the plasma membrane. And when lazy 3M cherry was ectopically expressed there in root epidermis, and lazy 3 was found on the plasma membrane, and GNOME GFP signal became obvious on the plasma membrane. So as a collaboration with Joan Don's group, we demonstrated that RLD3 physically interacts with GNOME. And in root epidermis, RLD3 is co-localized with GNOME, specifically obvious co-localization, especially the obvious co-localization at the plasma membrane was found. You can see here. And recently we found that GNOME GFP on the plasma membrane became almost undetectable in the RLD1, 2, 3 triple mutant background. These are rather preliminary data, but GNOME might be recruited to the plasma membrane by RLD function. So we don't know whether the same can be said for corneal cell, but RLD is recruited to the plasma membrane by lazy function, and this RLD might gather other factors involved in pin regulation, such as GNOME and possibly D6PK, as indicated by Yuji's group. And we suppose that the auxin transport regulatory platform formed here might be transiently formed on the plasma membrane through the lazy function. And our research provided a molecular framework of gravity sensing and signaling, and this working hypothesis fits very well with a position sensor hypothesis proposed by Yoel. So finally, I'd like to thank our lab members and collaborators, Takeshi Nishimura and Shogo Mori, and Hiromasa Shikata greatly contributed to this work. And Takeshi and Hiromasa and Nozomi Kawamoto are presenting posters, so please stop by their poster if you are interested. So that's all. Thank you for attention.